Hi, Jean. You here to listen to my presentation?
Yes.
Hi.
Hi. How
Hi.
are you?
I'm good. Can you please type your phone name in the chat just to take attendance? And
Oh, sure.
I'll begin in just a moment.
Mhm.
Let's see. Okay. All right. So welcome to my presentation. My name is Christina Nguyen, as you already know, and I am presenting to you this poster testing pine oil as a biofriendly substitute for xylene in histological staining techniques. I work on this project with Navne Arzu Varogi, Taylor Nguyen, and our research mentor for this project is Dr. Adriana Patricia Bisbal. And now I'll give you some background information to better understand our project. All right, so to better understand our project, as you can already tell from the title that I work with histological stains, specifically hematoxylin and eosin, also known as H&E for short, and mixed trichrome staining. H&E is just a general type of stain in histology. And then there's the Mason's trichrome. Mason's trichrome is a special type of stain in histology used for differentiating collagen fibers and muscle fibers. Both of these type of stains, they are for visualizing tissue structures. All right, so for the histological staining process, there's this reagent called xylene that histologists use. What does this reagent do? It dewaxes, which is the removal of paraffin off of the tissues, and it also clears tissues in order for us to add dyes and stuff like that. These are in the deparaffinization and dehydration steps in histological staining techniques. Unfortunately, xylene is toxic and non-environmental friendly, which is why histologists, they research biosafe agents to substitute xylene in histological stains such as H&E. And for this project, we picked pine oil. And this research question for the project, can pine oil effectively substitute xylene in deparaffinization and dehydration steps in H&E and Mason's trichrome staining? I will now show you figure 1. This is just a schematic to show you how we did our project. So the first step, we have the formalin fixed paraffin embedded mouse mammary gland tissues. These were sectioned by the microtome. After we sectioned the tissues, we then mount these tissues on slide and then randomize them to prevent bias. And then we remove the paraffin off of the tissue, which we call the deparaffinization step. And here's this nice chart right here, the deparaffinization step. What reagents we use will depend on the experimental group on the first column. So for example, xylene, that means I'm going to use xylene in the deparaffinization and dehydration and clearing step. All right, so after the deparaffinization step, we have the histological staining step, which is when we add colors to these tissues. So in H&E, we add hematoxylin, which will stain the tissue purple, and then eosin will stain the tissue pink. There's only 2 colors in H&E, and then for the Mason's trichrome, you will see red, blue, and purple colors. After the histological staining step, we then move on to the dehydration and clearing steps. This is when we, once again, depending on which experimental group it was, we either use xylene or pine oil. And then after this step, we then have the mounting of the tissues, add the cover slip to the tissues, use the microscope to take a look at the tissues, and then finally we image them. And now I'm going to show you part 1 of my results. This is figure 2, in which I did a quality comparison of xylene and pine oil in the deparaffinization and dehydration steps of H&E staining. The larger images, those were taken at 40x total magnification, and then the little image on the corner, those were taken at 400x total magnification. Since this is H&E, we only have pink and purple for this type of stain. So once again, the purple color, that's because we have hematoxylin, and that stains the nuclei and basophilic structures such as a lymph node, like this big blob here, that's a lymph node. And then the pink colors that you're looking at from this these images, those are because of eosin, which only stains the extracellular matrix. And if there's blood cells on the slide, those might be stained pink as well. So how do we determine the quality of the stains by looking at these images for each group? Well, we determine it by the clearness of tissue structures, meaning do I see any distortion or blurriness in the photos, or are the tissue structures clear for me to identify? The next category, we have color intensity, how dark or light were the tissue stain for these images. And then color balance, since this is H&E, I want to see a balance between pink and purple. That's the ideal condition, but a bad color balance would be like if there's too much pink that you can't see the purple, and vice versa. And then lastly, when we look at the mammary gland ducts, shown in these small images right here, those are mammary gland ducts. I should be able to see nuclei which are stained purple in H&E. So going through each of these groups one by one, here's xylene. The images in this group right here, you can see that there are no distortions or blurriness in these two photos. Based on the color intensity, they both look darkly stained. And for the color balance, since they're both of the colors are stained dark, there's no color balance issues. And then lastly, because I look at this mammary gland right here, the nuclei are easily identifiable as you see these little purple dots. Moving on to xylene pine oil, we have pretty much the same result like this one in xylene. So there's no distortion or blurriness. Color intensity, both colors are dark. Color balance, pretty much good because none of the colors look like they're overpowering one another. And then in the mammary gland duct, we can see the nuclei, which are shown in this image right here, the dark purple dots, those are the nuclei. I can distinguish them. However, moving on to the pine oil xylene group, that's when we start seeing color intensity issues. So if you look at this overall image right here, notice how like it looks faint compared to these two up here. And then for the color balance issue, although this image looks faint, I could still see the lymph node stained dark and somewhat purple, and then some part of the ECM, but it's not distributed evenly across the tissue. However, by looking at this mammary duct right here, I can still distinguish the nuclei, which are the little purple dots in this mammary gland duct. So that's still good. And then the last group, pine oil. Pine oil, once again, there's color intensity issue because the tissues were stained faint. And there's also a color balance issue because there are some areas that are like more darker than the others, though I wish they're spread more evenly in terms of color intensity and color balance. But otherwise, I can still see the nuclei just fine in this mammary gland duct right here. So what can I conclude from figure 2? Well, none of the groups appear to have clearness issue, and they also have distinguishable nuclei in each mammary gland duct. But for the groups that use pine oil in a deparaffinarization step, there were color intensity issues as shown in these two images here, and there were also some color balance issues. All right, moving on to figure 3. This is a quality comparison of xylene and pine oil in the deparaffinarization and dehydration steps of Mason's trichrome staining. Mason's trichrome staining, I should remind you again that this is a special type of stain used to differentiate collagen from muscle fibers. So we have 3 colors in Mason's trichrome. We have red, purple, and blue. So the red areas, those stain the muscle fibers. The purples, those are the nuclei, and the blue shown nicely here, those are the collagen. And once again, the magnification are the same as the one from H&E. So images, the larger images, those were taken at 40x total magnification. And then the images on the corner of mammary gland ducts, those are just 400x total magnification. So starting with the xylene group, we can see a nicely dark stain of the muscle fibers shown in dark red. We have some collagen distributed throughout the tissue as well. So those blue areas are collagen, including this one right here in this mammary gland duct. And then here are the nuclei stained purple. There doesn't appear to be any color balance issue. All the three colors are stained nicely dark as well. And there's no distortion or blurriness in this photo. And I can distinguish the nuclei in this duct. Moving on to xylene pine oil group, here are the muscle fibers in red, collagen in blue, and then we see the nuclei in purple here. Unlike this group right here, we have the mammary gland duct appears less saturated, so this tells me that there must have been a color intensity issue for this group in particular. But otherwise, for the color balance, it is fine. We can see all three colors. Then we have pine oil xylene. Here's the muscle fibers in dark red. Here's collagen blue, and then nuclei purple. This group in particular looks similar to this group up here, xylene, in terms of like saturation. And then the last group, pine oil. Here's some muscle fibers, those dark red areas. And then if you look at this one in particular, this is the extracellular matrix. This one's stained dark versus the one right here. So this tells me there's also color intensity issue in this group. And that's the same thing for this mammary gland duct. These were the nuclei and the collagen were stained faint like the ones up here. It's not like these two were saturated. So based on according to, sorry. So for figure 3, I conclude that the groups that use pine oil in the dehydration and clearance step, those were having color intensity issues, but otherwise, all the images look clear, and the pine oil is good at removing the paraffin off of the tissue. You move to 4. All right, and this is figure 4. I have two different data because I did stainings in 2 different semesters, and the purpose of this figure 4 is to show you the quality comparison of xylene and pine oil H&E staining over time. I would like to let you know that the June 2021 data, we only use old reagents for this one because at the time we didn't have access to new reagents. So stuff like hematoxylin and eosin, we had to pretty much recycle them during June 2021. Whereas the December 2021, we finally have new reagents, so we don't need to do that. Images at the top, those were taken at the time that we did the initial staining, and then the ones on the bottom is just to show you what did it look like after a certain amount of time has passed. So June 2021, here's xylene. We can see some pink and purple in this mammary gland duct. And then for the pine oil, there's barely any pink, but there are some red blood cells that are slightly stained pink, but it's kind of barely visible. Then we have the hematoxylin, which are the purple areas. Those were stained. You can see purple here in this mammary gland duct. 6 months later, we see that the pink fades away, and you cannot see this at all in pine oil. So this led to me conclude that after 6 months, the eosin quality fades away. And then now for the December 2021 data, here's xylene and pine oil. We both have pink and purple for both of these groups. And just after 2 months, they're still there. So it's still a bit early to conclude if the age of the reagents have an effect on on the tissues colors, or does this have to do with the protocol, or is it because I use pine oil in H&E staining? To summarize my project, so we determined that pine oil is a suitable alternative to xylene for the deparaffinization steps and adequate for dehydration and clearing steps of histological staining techniques. We also conclude that pine oil treated histological stain had color balance and color intensity issues as shown in the previous figures. And then last but not least, of the summary, we have the eosin quality fades away at 6 months for both xylene and pine oil treated stains. And once again, it's still a bit too early to conclude if it has something to do with how old the reagents were, or was it our protocol, or is it because I used pine oil in the stains. So for future directions, we would like to optimize both H&E and Mace's trichrome staining protocols with pine oil and testing other tissue types such as kidney or lung. Because this project, I only use mammary gland tissue. That's why I want to test this on kidney and lung. We also want to test pine oil in other downstream histological applications such as immunohistochemistry. And then last but not least, recruit histologists to evaluate the quality of pine oil in histological stains. Here are my references, and I would like to acknowledge DOED and MSEIP for partially funding this project, as well as acknowledge other contributors to this project, Histology Lab from spring 2021, as well as Berkeley Garza, Onaji Bloom, and Gabriela Villalobos, and I thank you for listening. And I will now take any questions, if anyone has any. Will I be continuing the research? I would like to, but I'm graduating next month, so no, I'm not continuing this research. Oh, and hello, anyone who came in late. I will give you a overall view of my research in just a moment. Let me just address the questions if anybody has any. And please enter your name in the chat so you haven't.
Oh, yeah, I'm sorry. I wanted to ask what exact differences you expect to see in kidney and lung tissue?
Well, because these organs, they're made of different tissue compositions, I expect to see like there might be like a difference in the color intensity of how they stain the tissues. But I'm ideally looking for if pine oil can give me the exact same results as xylene and histological stains. I don't know if that answers your question. I want to see if pine oil will work in other tissues.
I see, that makes sense. Thanks.
Yes. Any other questions?
Yeah, one question. For the xylene pine oil and then the pine oil xylene, what's the difference between those two? Is it the order you use them in?
You're talking about the groups in general, right?
Yeah.
So yes, that will depend on the experimental groups I showed you earlier from figure 1. Can you see this, Jean? So
yes.
So if I say like xylene, that means I used it in the deparaffin and the dehydration and clearing step. And then xylene pine oil, I use xylene in the deparaffin step and pine oil in the dehydration and clearing step. Does that make sense, Jean?
Yes.
It's like what you're doing right now in histology, actually. All right, I think that's my visitors from previous section. Is that all your questions, Jean? So I can present to the next two people?
Yes, it was good job. Thank you.
Thanks, Jean.
Bye.
Thanks, Jean. All right. All right. Hi, everyone, for joining my poster presentation. I'll just get started in a moment if I can move my poster. Yeah. All right. Did everyone sign in? Okay. All right. So welcome to my poster presentation. My name is Christina Nguyen, and today I'll be presenting to you testing pine oil as a biofriendly substitute for xylene in histological staining techniques. I work on this project with Namit Arzu Forogi and Taylor Han Nguyen, and our research mentor for this project is Dr. Adriana Patricia Visval. All All right, to better understand the project, I will now give you some background information. So as you can tell from the research title of this project, I work with histological stains. And for this project in particular, I am working with hemotoxin eosin, also known as H&E for short. And then the other stain that I did in lab was Mason's trichrome. So hemotoxin eosin is just a general type of stain used by histologists just to see the just to visualize tissue structures. But Mason's trichrome is a type of special stain in histology, and it's used for visualizing differentiating collagen and muscle fibers. In histology, we have this reagent called xylene. It's used in histological staining techniques to de-wax, which is the removal of paraffin off of the tissue, and it's also used to clear tissues. Both of these occur in the deparaffinization and the dehydration steps of histological staining processes. Unfortunately, histologists, I know that xylene is important in the histological process, but it is toxic and not environmental friendly, which is why histologists, they are testing biosafe reagents to substitute xylene in the histological staining. And for this project, we work with pine oil, and the research question is, can pine oil effectively substitute xylene in deparaffinization and dehydration steps of H&E and Mason's trichrome staining? And now I'm going to show you figure 1. So we have the formalin-fixed paraffin embedded mouse mammary gland tissues. We section these with a microtome. After that, we take these samples and mount them on slides, and then we randomize them to prevent bias. After that, we then remove the paraffin, which we call the deparaffinization step in histology. And depending on which experimental group shown on the first column, this will tell us, am I going to use xylene or pine oil for the deparaffinization step? And it's the same thing for the dehydration and clearance step. This will depend on the experimental groups. But back to the steps. So after the deparaffinization, we then do the histological staining, in which we either do H&E or Mason's trichrome. H&E will give the tissue pink and purple color, whereas the Mason's trichrome will give you the red, purple, and blue colors. So after we colorize the tissue with the histological staining, we then subject the tissue slides to dehydration and clearing step, in which we either use xylene or pine oil, depending on the experimental group shown here. And then the last steps, we have mounting of the tissues, and we add a cover slip. Then we have to use a microscope to take a look at these tissues, and then we begin imaging. And now I'm going to show you figure 2. This is part 1 of my results. So here's quality comparison of xylene and pine oil in the deparaffinization and dehydration steps of H&E staining. The large images, those were taken at 40x total magnification, and then the images on the corners, those were taken at 400x total magnification, just to show you what a mammary gland duct looks like in the tissue. And how do we grade the quality of the stains? We determine it by the clearness of the tissue, meaning, do I see any distortion or blurriness in the tissue, or are the tissue structures clear for me to identify? Then we have color intensity, in which we ask ourselves, how dark or light do the colors stain the tissue? Color balance, since this is H&E, we want to see a balance between pink and purple. So let's say, like, if the pink is stained dark, and then I want the purple to be like the same intensity as well. Hopefully none of the colors overpower one another, because if that's what happened, then we won't be able to see the other color. So if we see the pink being too overpowering, we won't be able to see the purple. And then last but not least, in the mammary gland ducts, we should be able to distinguish the nuclei, which are those itty bitty dots stained purple in the mammary gland duct images. Oh, I forgot to say that hematoxylin, it will stain the basophilic structures such as lymph node, and they will also stain nuclei. And then the eosin that stains the extracellular matrix, which will give it a pink color. I will now go over the groups one by one, based on the criteria that I listed earlier. So xylene, we can see that there are no distortion or blurriness in the photos shown here. And then as for color intensity, both pink and purple stain dark. And since they're both the same color intensity, I would say that there's color balance in this group. And you can see the nuclei in the mammary gland duct shown here, which are the ones in purple. Xylene 0.0 has about the same result as xylene group. So there's no blurriness or distortion issues shown in these two images. Color intensity, those were stained dark. And then color balance, since both of the pink and purple are stained dark, then we have color balance. And then last but not least, the mammary gland duct, we can see the nuclei stained purple. I can distinguish them. Moving on to the xylene group, that's when we start encountering color balance and color intensity issues. So for the color intensity, notice how like these this overall image looks faint compared to like these two up here. And as for the color balance issue, the lymph node over here, I can see that they're much darker than like other areas that's supposed to be stained purple. And then we have pink going on right here as well. Like this area is stained dark, but not like in these areas. So color intensity issue and color balance issue. But looking at this mammary gland duct, we have the nuclei stained purple, and since I can distinguish them, that's good. There's no clearness or distortion issues. There's no distortions or blurriness in these two images. And then finally, pine oil. We have no blurriness or distortion in the images, but we have color intensity and color balance issues. So notice how like it looks similar to this group right here. So we have fading of the pink and the purple, and for some reason the color balance is also not good. We have some areas stained much darker than the other parts of the tissue. But looking at this nuclei right here, we can see that the purple stain the nuclei nicely, and I can distinguish them with this mammary gland duct. So for this figure, I conclude that the pine oil that was used in that depigmentization step, they had color intensity and color balance issues, but otherwise, overall, all these did not suffer from distortions and blurriness in the stains, and we also can see that they all have nicely defined nuclei shown in the mammary gland duct. Moving on to figure 3. This is a quality comparison of xylene and pine oil in a deperiodization and dehydration steps of Mace's trichrome staining. Once again, I should remind you that Mace's trichrome is a special type of stain to differentiate collagen fibers from muscle fibers. The larger images, those were taken at 40x total magnification, and in the images on the corners, those were mammary gland. Those were images of the mammary gland ducts, and these were taken at 400x total magnification. How do we grade the quality of Mace's trichrome staining for each group? That's once again like how we did the H&E. So we determine how clear do the tissues look? Do we see any distortion or blurriness in the images? And color intensity, how dark or faint were the tissues stained by the dyes? Do they appear too dark or do they appear too light? Color balance, do we see an equal distribution of those three colors, or do one or two appear more overpowered than the other, that we can't distinguish the other colors? And then last but not least, when we look at the mammary gland duct, can we distinguish the nuclei? So going by this figure, each group one by one, starting with xylene. Here is the muscle fibers in dark red. And then these light blue areas, including the blue right here, those are the collagens. And then the purple in this membrane duct right here, those are the nuclei. We don't have any clearness. We don't have any distortion or blurriness in this group right here. But other than that, nice color intensity. All the colors appear dark, and we have good color balance. And we can distinguish the nuclei in this group. Moving on to xylene pine oil, here's the muscle fibers stained dark red. Here are some collagen out there in blue, and then this is the nuclei. But notice how this mammary gland duct appears less saturated than the ones right here. This tells me that there must have been a color intensity issue in this group in particular compared to xylene. But otherwise, we have nice color balance. Then we have pine oil xylene. Here's the muscle tissue. Muscle fibers stained red. Here are the collagens in blue, and then this is the nuclei in purple. And this looks identical to this group up here, xylene. And then last but not least, pine oil. Here's some muscle fibers. We can barely see collagen stained blue in this group in particular. And here's the nuclei in purple in this mammary gland duct. Notice how this side is much darker than the ones right here. So this tells me that there must have been color intensity issue for pine oil because they appear faint. And this reminds me of this image right here shown in the xylene pine oil. These two groups use pine oil in the dehydration and clearing step. So this tells me that using pine oil in that particular step for this type of stain, they suffer from color intensity issues. But otherwise, all the groups, they all had good clearness, and they all have distinguishable nuclei in the mammary gland ducts. And here's my final part of the results, figure 4. What I'm doing right here is quality comparison of xylene and pine oil H&E staining over time. I have 2 different data because I did multiple stainings throughout the research. So here's the June 2021 data, and then here are the December 2021 data. I do want to point out that I use overagents on this one, meaning that I recycled the reagents in the histological stain because we couldn't get any new reagents. So that's what I want to point out. And then for December 2021, we finally got new reagents, so I was able to use the reagents without having to reuse them. All right. So images on the top, those were taken when I first did the initial staining, and then the images on the bottom just to show you how do the stains look over time. So starting with June 2021, here are xylene and pine oil images that I took initially when I first did the staining. Notice how xylene I have both pink and purple shown here, and then pine oil, there's barely any pink. Those little itty bitty dots right here, those are the red blood cells. Normally they're stained dark pink, but they only appear faint in this one. 6 months later, the pink fades away for both of these groups, and you cannot see pink at all in this group right here. But otherwise, the purple areas in both of the groups, they remain after 6 months. So this tells me that the eosin or the pink for H&E staining, whether it be for the xylene or the pine oil group, they fade away after 6 months, but the hematoxin remains there. So now we're moving on to the December 2021 data. We have both pink and purple for both of the groups stained nicely. And after 2 months, neither of the colors fade. So although that you see there was fading in the June 2021 data, it's still a bit too early to determine if the fading of the pink was because of how old the reagents were, or it has something to do with our protocols, or it could be because I used pine oil in the H&E staining. I would need more data on this in order to conclude if any of that are the reasons for the fading of the pink. So to summarize my project, we determined that pine oil is a suitable alternative to xylene for the deparaffinization steps and adequate for dehydration and clearance steps of histological staining techniques. We also saw that pine oil treated histological stains had color balance and color intensity issue, as I have shown you in previous figures. And then the last summary, yields and quality fades away at 6 months for both xylene and pine oil treated stain. But once again, we don't know if it has to do with how old the reagents were, or if it's because of our protocols, or is this because I use pine oil in the stain. And for the future directions, we would like to optimize both H&E and Mace's trichome staining protocols with pine oil and testing other tissue types such as kidney and lung, because I only work with mammary gland for this project. We would also like to test pine oil in other downstream histological applications such as immunohistochemistry. And last but not least, recruit histologists to evaluate the quality of pine oil and histological stains. Here are my references, and I would like to acknowledge DOED and the CIP for partially funding this project. And I would also like to acknowledge other contributors to this project, Histology Lab for Spring 2021, as well as Bertha Garza, Onaje Blunt, and Gabriela Villalobos. And thank you for listening. I'm happy to take any questions.
Great job, Christina. Dr. Parker, you're muted.
Yes, go ahead. Do you have a question?
Yes, I do have a question. Yeah, I just wanted to ask, I know you talked about using the, the intensity of the colors on the tissues to figure out, your results, right? So I was just wondering, do you have a scale that you use to measure the intensity, or do you just like eyeball it? Because, sometimes you need to have a way to, quantify things in order to be consistent in the experiment.
That's a good question, Price. Thank you for that. We actually are constructing a rubric right now to how are we going to grade the histological stains, but we're only eyeballing the intensity of the colors. We don't have a scale to determine is this good enough or is this bad?
Do you think you could use like an absorbance, be it maybe a reflective, I forget the names of the machines, but anyway, I wonder if that could work.
That is what I don't know. I might have to talk with Dr. Vispa about this.
Yeah.
Mm-hmm.
Well, the other thing is I wanted to know now, is pine oil less toxic or dangerous than the other? Is that why you're studying both of those? I'm just curious.
Yes. Pine oil is a biosafe reagent, and we're just trying to see, can I get the same results as the xylene, which is the toxic reagent. Right. I want to see if I can use pine oil in these stains to see maybe we can replace xylene in the histological staining process and make it safer for the histologists to use. That way they don't need to use xylene.
Yeah, that would be great. And actually, using the paraffin, I mean, I'm just personal question here. Did you find it difficult to shave it at a microtome level that needed to be? How were you on that? I'm just curious, since I've done it myself.
Oh my gosh, that was really annoying. Having to cut these tissue with the microtome, and then I had to clean up every time I used it in the lab. All the paraffin shavings are all over the place, Dr. Parker. All over the microtome, all over the counter. I'm like, oh no.
Oh, I know, girl. And I was doing that with Drosophila salivary glands, and just trying to get the gland out was killing me. And then we attacked the paraffin with the microtome. It was a great lesson in patience, is what I will say. Well, thank you. And thank you for recording this. I love that you're doing this type of study. And how was it received at TAS?
Oh yeah,
in your opinion?
Tell you this. I think according to Dr. Vispa, out of all the posters that were I think in my category for biology, I can't remember how many were there, maybe over 20 people in biology, they presented a poster too, but I scored like either 4th place or 5th place out of 20 something posters.
Oh my gosh, that's great. That's great.
And
Wonderful.
I don't remember exactly, but I think like Dr. Viswell said, I'm 2 points away from 3rd place.
Oh, that's amazing. And was that your first conference
Yes,
presentation?
first conference.
Look at you. Well, keep at it is what I'll say, right? Okay, I'm going to excuse myself. I've got others to see. Continue. And thank you all for coming to our presentation.
Thank you, Dr. Parker and everyone.
Thank
Catch
you.
my breath.
Okay.
Hello, sorry, I just came when you guys was done. Sorry.
It's okay. I just need to catch my breath for a moment.
That's
and
okay.
then I'll go over the project with you.
That's okay. Take your time.
Have you signed in?
No, I have not.
Just type your name in the chat and then that'll do. And I'll get with you in just a moment. Alright, let me set this up. All right, so thank you for visiting my booth. I will now explain to you my project. So welcome, my name is Christina Nguyen, and my project is testing pino as a biofriendly substitute for xylene and histological staining techniques. I work on this project with Nagmet Arzu Furogi and Taylor Han Nguyen. And our research mentor for this project is Dr. Adriana Patricia Viswell. Excuse me. I will now introduce you to my project, give you some background information to better understand it. All right, so as you can tell from the research project title, I work with histological stains, specifically hematoxylin eosin, also known as H&E for short. And the other stain I work with was Mace's trichrome staining. So H&E is just a general type of staining used by histologists, and then Mace's trichrome is a special type of stain. It's used for differentiating collagen and muscle fibers. Both of these type of stains, they are used for visualizing tissue structures. So in the histological staining process, we have this reagent called xylene. This is a reagent used for dewaxing, which is the removal of paraffin off of tissue samples. And it's also used for clearing tissues. Both of the dewax and the clearing of tissues are in the deparaffinization and dehydration steps. Although xylene is important in the histological staining process, unfortunately it is toxic and not environmental friendly, which is why histologists, they are testing biosafe reagents to substitute xylene in histological stains. And for this project, I chose pine oil as a xylene replacement. And the research question for this project: can pine oil effectively substitute xylene in deparaffinization and dehydration steps in H&E and Mace's trichrome staining? And now I'm going to walk you over figure 1. This is just a schematic design of H&E and Mace's trichrome staining techniques. So the first step of my project, first we had the formalin fixed paraffin embedded mouse mammary gland tissues. We section these with a microtome. And then after that, we take these tissue samples to be mounted on slides and then randomize them to prevent bias. After that, we then remove the paraffin. We call this the deparaffinization step, and this will depend on the experimental group shown in the first column. So for example, this group says xylene, that means it's telling me I'm going to use xylene in the deparaffinization step and dehydration and clearing step, which I'll get to in just a moment. After the deparaffinization step, we had the histological staining step, which is when we use H&E or Mace's trichrome. H&E, this will give the tissue pink and purple color. The purple color comes from hematoxylin, and then the pink color comes from eosin. Mace's trichrome, you will see red, blue, and purple. So after we did the histological staining, which will colorize the tissue, we then move on to the dehydration and clearing step. And for this step, once again, it will depend on the experimental group we have here. I either use xylene or pine oil, depending on which experimental group it is. And then the final steps, I mount these tissue, put on the cover slip, put them on the microscope, take a look at them, and image them. And then I will now show you part 1 of my results. All right, so figure 2. This is a quality comparison of xylene and pine oil in the deparaffinization and dehydration steps of H&E staining. The images that are big, those are just the overall view of what the mammary gland looks like. And these were taken at 40x total magnification. And then these images shown on the corners, those were taken at 400x magnification to show what a mammary gland duct looks like up close. So how do we grade the quality of the H&E staining? Well, we determine it by the clearness of the tissue, meaning do I see any distortion or blurriness in the photos, or do the tissue structures appear clear enough for me to identify them easily? Then the color intensity, how dark or light did the color stain the tissue? Then we have color balance. Are the pink and the purple balanced in the tissue, or do I see one of the colors that overpower one another? What do I mean by that? Well, let's say the pink is too dark, then I won't be able to see the purple in the tissue and vice versa. We don't want that. We want to see equal intensity and distribution of pink and purple for H&E. And then last but not least, when I look at the mammary gland ducts, I need to be able to distinguish the nuclei, which are stained purple, these little itty bitty purple dots. And now I'm going to go over each group. So here's xylene. We see that there's no blurriness or distortion in this these two images. Color intensity, the colors look like they're stained dark. And then as for the color balance, because they're both of the colors are stained dark, and we do have a color balance between pink and purple. None of them overpower one another. And then last but not least, in the mammary gland duct, you can see that we have nuclei stained purple. I can distinguish them, so that's good. Xylene pine oil has the same results as this group right here, so no blurriness or distortions in the images. Color intensity for both colors, they're both dark. Color balance, yes, there's color balance between pink and purple since they're both of the same intensity. And then here are the nuclei in the duct stained purple. Moving on to pine oil xylene, that's when we start to see color intensity and color balance issue. What do I mean by that? Look at this overall image of the mammary gland tissue. This shows that the colors pink and purple appear faint, unlike these two up here. And what do I mean by color balance issue? This purple area right here, like I can see it stained darker, but then like the pink is like mostly faded. So I wish like the pink was the same intensity, but since we don't have that, we do have color balance issue. But otherwise, the nuclei in the mammary gland duct, they are stained purple, and I can distinguish them, so that's good. And then pine oil group, once again, there's fading issue and in the color intensity, and also color balance issue like this group right here. But otherwise, there are no distortion or blurriness in these photos, and I can also distinguish the nuclei shown in this mammary gland duct. So what can I conclude from figure 2? I can conclude that the pine oil, when it's used in the deparaffinization step, that we start to see color intensity issue and color balance issues, but otherwise, pine oil is it shows that there's no distortions or blurriness, and also you can distinguish the nuclei pretty much just like the xylene group here. So now we're going to move on to figure 3, which, well, I will be talking about Mace's trichrome results. So figure 3, here's the quality comparison of xylene and pine oil in the deparaffinization and dehydration steps of Mace's trichrome staining. Once again, these large images I show right here, these were taken at 40x total magnification. And in the images on the corners, those were taken at 400x total magnification, just to show you what a mammary gland duct looks like. And let me remind you that in Mace's trichrome, we use a special stain to differentiate collagen and muscle fibers. So the muscle fibers are the ones stained in dark red, and then The collagen fibers are the ones stained in blue. And in the purple areas, that's just the nuclei. So in xylene, we can see that there is clear structures everywhere. There's no blurriness or distortion for this group. The color intensity, it looks like all three colors, they stain dark and nicely. And there's color balance between the three colors. And then the last thing, here are the nuclei in the duct. I can see them. Xylene pine oil, here's the muscle fibers staining dark red, nice. And we also can see there's light blue and then the purple nuclei here. But unlike this group right here, notice how that this mammary gland duct appears less saturated than the ones right here. So this tells me that there must have been color intensity issue going on in this group when we use pine oil in the dehydration and clearing step. But otherwise, the tissue appears, does not appear blurry, does not appear distorted, and I can still distinguish the nuclei just fine. Pine oil xylene, here's the muscle fibers in dark red. Here's the collagen in blue from this mammary gland duct, and in the purple area, that's the nuclei. This looks identical to this group right here, xylene. There's no color intensity issues, nor color balance issues. There's also no distortion or blurriness in these photos. And then the last group, pine oil. Here's some muscle fibers. And notice how this area of this tissue appears darker than this side. So this tells me that there's color intensity issue in this tissue. And there's also a color balance issue as well. The dark red appearing more saturated than the blues and the purple. And like this mammary gland duct right here is just identical to this group up here, xylene pine oil. And once again, the pine oil, if it's used in the dehydration and clear step for Mason's trichome, we have color intensity and color balance issues. But otherwise, there's good clearness, and there's also good distinguishable nuclei in each mammary gland duct. And then moving on to the final figure for this poster, figure 4. This is just a quality comparison of xylene and pine oil H&E staining over time. All these images are of a mammary gland duct, and they were taken at 400x total magnification. I would like to point out that in the June 2021 data that I have right here, I only use old reagents because at the time we don't have new reagents, so I had to reuse and recycle the reagents for this H&E staining. The December 2021 data, though, those were when I finally got new reagents, so I don't need to recycle or reuse the reagents, and yeah. Images on the top, these images were taken initially when we first did the staining, and then the ones on the bottom is a certain time has passed since the initial staining, just to see how do the H&E look over time. So paying attention to June 2021 data, here's xylene and pine oil. You can see both pink and purple just fine as xylene, but notice how in this group right here, pine oil, we have barely any pink going on, but there are red blood cells right here, lightly stained pink, but otherwise it's mostly absent. The purple is still there though. 6 months later, the eosin, the pink fades away from both groups. This one does not have any pink at all anymore. So this led me to conclude that after 6 months, the pink fades away. And then here's the December 2021 data. Here's the initial images. Sorry. Here's the initial images, and then 2 months later, we still have pink and purple. So what can I conclude overall for figure 4? Well, it's actually a bit too early to conclude for figure 4 in general, because we don't know for sure if the age of the reagents are the cause of the pink to fade away, as shown in this data right here. Or number two, it could be because of the protocol. And then three, it could be because I use pine oil and H&E. So it's still too early to include. I need more data to properly conclude the quality comparison of xylene and pine oil H&E staining over time. And to summarize the points of my research project, we determined that pine oil is a suitable alternative to xylene for deparaffинization steps and adequate for dehydration and clearance steps of histological staining techniques. Pine oil treated histological stain had color balance and color intensity issue as shown in previous figures. And then the final summary point is that use and quality fades away at 6 months for both xylene and pine oil treated stains. But then once again, we don't know if it's because the age of the reagents caused the fading, or it could be because of my protocols, or it could be because I use pine oil in the H&E stain. So future directions. We would like to optimize both H&E and Mace's trichromacy staining protocols with pine oil and testing other tissue types such as kidney and lung, because I only work with mammary gland for this project. So might as well test this on other tissue types. We would also like to test pine oil and other downstream histological applications such as immunohistochemistry. And then last but not least, recruit histologists to evaluate the quality of pine oil in histological stains. And here are my references. I would like to acknowledge DOD and MSCIP for partially funding this project. And I also like to acknowledge other contributors to this project, Histology Lab for Spring 2021, as well as Bertha Garza, Onaje Bloom, and Gabriela Villalobos. And thank you for listening. Do you have any questions? That's a mouthful. Chat. What motivates you to look at pine oil rather than other alternatives? Well, to answer that question, this started off as a project in histology lab course, and this project we had to read papers published by other scientists. What bioreagents are there that we can use to substitute xylene in histological stains, and my group back in histology lab last year, we found this paper where this group of scientists used pine oil, and we decided to test that. These group of scientists only use pine oil in the clearing steps of H&E, but they never used it to deparaffinize tissue. So that's what we tested this for. And I became obsessed with this project after I finished histology lab, and I asked Dr. Viswell if I could stay with her to keep working on this, and that's how I end up here 3 semesters later. I'm still in Dr. Viswell's research, and right now I'm trying to wrap everything up. And you can see that the Mace's trichome, that's actually pretty new because I just started that last semester. I hope that answers your question. Histology lab is very fun. That's my motivation. And also because we read papers. Xylene hazardous. I would say the exposures to this reagent, I read it from one of the papers I referenced in my poster, that, just to paraphrase it, they discovered that it can cause many things like skin irritation, some nerve damage, and some other nasty things that xylene does. So that's why we are trying to substitute xylene in histological stains and hopefully make it safer for histologists to do stainings. That way they don't have to use xylene. Use biosafe reagents such as pine oil, which is what I just presented. Hope that answers your questions. Sorry, I'm not familiar to most of these subjects. What does inconsistent? Well, to answer that question, it depends on what type of stain it is. So, for example, H&E is a general type of stain. Histologists just use this to analyze tissue structures, and for that, they will see pink and purple. And then Mason's trichrome, that's used for differentiating muscle fibers and collagen. That's just some examples of histological stains. There's a lot out there. And does that answer your question, Bianca? Right? Yeah, Bianca. All right. Thank you for visiting my booth. Oh, and hi, Karina. Welcome, welcome.
Hi. Thank you.
Sorry, I just need to catch my breath. I've been talking like the entire booth.
No, you're good. You're good. Take your time. I'm here. I've heard it before, but I'm still here to hear it again.
It's okay. I'll go over it after I catch my breath. I presented to a lot of people. I'll go over it again in just a moment. So just to be clear, xylene is used in labs that study tissue samples. Yes, in histology we do use xylene. It's required to actually, because without it we can't really stain tissues. Oh, and Karina, can you please type your name in the chat just for me to take attendance? That you
Yes,
visited my booth?
I got you. Thank you.
I'll start in just a moment. All right, so here we go again. Hi, my name is Christina Nguyen, and I am presenting to you testing pine oil as a biofriendly substitute for xylene histological staining techniques. I work on this project with Namen Arzu Farogi and Taylor Han Nguyen, and our research mentor for this project is Dr. Adriana Patricia Visbal. All right. All right, so for the background information to better understand our project, as you can tell from the title, I work with two different types of stains in this project. The first is hematoxin eosin, also known as H&E for short. This is just a general stain used by histologists just to visualize tissue. There's another type of stain that I work with in this project as well called Mason's trichrome. It's also used for visualizing tissue structures. But Mason's trichrome, it is a special type of stain. And this is used for differentiating collagen and muscle fibers. In the histological staining process, there's this reagent called xylene. And this reagent is used for dewaxing, which is the removal of paraffin off of tissue samples, and it's also used for clearing tissues in the deparaffinization and dehydration steps. Although xylene is important in the histological staining process, it's unfortunately toxic and not environmental friendly, which is why histologists, they are testing pine oil as a, testing many different types of biosafe reagents to substitute xylene in histological staining. And for this project, we chose pine oil. The research question for this project it is, can pine oil effectively substitute xylene in deparaffinization and dehydration steps in H&E and Mason's trichrome staining? And now I'm going to walk you over figure 1. So figure 1, this is just an experimental design of H&E and Mason's trichrome staining techniques. The first step of our project, we had the formalin-fixed paraffin-embedded mouse mammary gland tissues. We sectioned these with a microtome. And then after that, we take the tissue samples mounted on the slide. And then after they're mounted, we then randomize them to prevent bias. We then subject these to paraffin removal, which we call it the deparaffinization step. And depending on the experimental group shown right here, this will tell me, do I use xylene or do I use pine oil for the deperiodization step? After that, we then move on to the histological staining, which is the colorization of the tissues. And we either do H&E or Mason's trichrome. H&E will give the tissue pink and purple color, whereas the Mason's trichrome will give it the red, blue, and purple color. And then dehydration and clearing step. This once again depends on the experimental group shown in this column. This will tell me, am I going to use xylene or am I going to use pine oil for this step? The final steps, we then mount these slides, add the cover slip, and put them on a microscope to examine the tissue. And that's also when we do the imaging process. And now I'm going to walk you over part 1 of our results. So figure 2, this is part 1 of my results. This is a quality comparison of xylene and pine oil in the deperiodization and dehydration steps of H&E staining. The larger images, those were taken at 48x total magnification, and then the images on the corner of each group, those are images of a mammary gland duct, and these were taken at 400x total magnification just to see what the mammary gland duct looks like and some other components. So how do we grade the quality of xylene pine oil in a deperiodization and dehydration steps of H&E staining? We grade these based on the following criteria. So the first one being the clearness of the tissue, like did I see any distortion or blurriness in the images, or do the tissue structures appear clear enough for me to identify the structures? The second one being color intensity, how light or dark do the colors stain the tissue? The third being color balance, since H&E only uses 2 colors, pink and purple, I want to see a balance of both of those colors. If I see too much of pink, I won't be able to see purple and vice versa. We don't want that. We want to see an equal distribution of those 2 dyes. And then last but not least, when I look at a mammary gland duct, I want to be able to distinguish the nuclei, which are stained purple. Forgot to mention this earlier, but hematoxylin stains nuclei and basophilic structures such as lymph node purple, and then the pink color comes from eosin. Eosin only stains the extracellular matrix and red blood cells. All right, to walk over each group, we have xylene shown here. So based on this, I don't see any distortion or blurriness in the photos. The color intensity for both colors look dark, and since they're both with the same intensity, I would say there is color balance between pink and purple. And in this image right here of this mammary gland duct, you can see the itty bitty purple dots. Those are the nuclei. I could see them just fine. So that's good distinguishable nuclei for this group. Moving on to xylene pine oil, the results shown these 2 images looks the same pretty much as xylene. So no distortion or blurriness. Color intensity for both colors are dark. Color balance for pink and purple, because they're both dark, and there is color balance. And then I can see the nuclei shown here in the mammary gland duct. But then we start to see problems arising in pine oil xylene. The first being color intensity, because both pink and purple stain very faint in this image shown here. And we also have color balance issues because the purple looks more visible than the pink shown in the overall image. This would have been better had both of them are staying at the same intensity, and both of them are balanced out in terms of intensity, just like the ones up here. But otherwise, there's no distortion or blurriness in this particular group, and I can still distinguish the nuclei if I look at the mammary gland duct photo. So pine oil, pretty much the same issues as this group right here. Pine oil xylene, we have color intensity issues because the colors stay faint, and we also have color balance issue because the purple is still more visible than the pink. But there are no distortions or blurriness in pine oil. And as for the nuclei, the purple dots, I can still see them just fine. So yes, I can distinguish them. So that's good. So to conclude on figure 2, the groups that use pine oil in a deparafffinization step suffer from color intensity and color balance issues, but otherwise, the clearness look great, and also the nuclei in the ducts are distinguishable. And then we're moving on to figure 3, the quality comparison of xylene and pine oil in the deparafffinization and dehydration steps of Mason's trichrome staining. Once again, I should remind you that Mason's trichrome is a special type of stain used to distinguish collagen and muscle fibers. So the dark red areas, those are the muscle fibers, and then the blue areas, those are the collagen, and then once again, purple being the nuclei and basofilic structures. So how do I grade the quality comparison in Mason's trichrome staining? It's pretty much just like H&E staining. I need to compare the clearness of the tissue. Do I see any distortion or blurriness, or do I can I identify the tissue structures just fine? Color intensity, how dark or light do the color of the dyes stain? Color balance, since there's 3 colors in this one, I want to see all 3 balance out, because otherwise, if one of them overpower one another, then that's a bad color balance. And then finally, when I look at the mammary gland duct, I need to be able to distinguish the nuclei, which are in purple. So starting with xylene, here's my muscle fiber, dark red. Then these light blue areas, including the blue in this mammary gland duct, that's the collagen. And then here's the nuclei in purple. So we have good color intensity here, since the colors are staying nicely dark. And we also have good color balance. It doesn't look like any of the colors are overpowering one another. There's no distortion or blurriness, and I can distinguish the nuclei in the mammary gland duct. Xylene pine oil, here's the muscle fibers, and then here's some collagen and nuclei. But notice how this mammary gland duct looks faint compared to the more saturated one in this group. So this tells me that xylene pine oil suffer from a color intensity issue. But otherwise, image looks nice and clear. There's no distortion or blurriness. And I can still distinguish the nuclei just fine. But the color intensity should have been darker in the mammary gland duct. Then we have pine oil xylene. Here's some muscle fiber, collagen blue, and then the nuclei in purple. This looks similar to this group right here, xylene. And then pine oil, we have muscle fiber right here, collagen in blue, nuclei in purple. But pay attention to this part in particular. Notice how this ECM is much darker than the one on right here on this side. And including the mammary gland duct itself, this tells me that pine oil suffer from a color intensity issue, just like this group right here. When you look at the mammary gland duct, there's also a color balance issue because this one's a lot more visible, the muscle fibers, and these two should have been stained dark as well. And other than that, there's no distortion or blurriness in pine oil, and I can still distinguish the nuclei. So what can I conclude from figure 3? The groups that use pine oil in the groups that use pine oil, they suffer from color intensity issues and color balance issues, but otherwise the images are still clear, and you can still distinguish the nuclei in the mammary gland ducts. And finally, the last figure, figure 4, the quality comparison of xylene and pine oil H&E staining over time. I would like to point out that for the June 2021 data, we didn't have access to new reagents, so I had to reuse and recycle pretty much the reagents at that time. Whereas December 2021, we finally managed to order new reagents, so I don't need to do any of that. All these images are of mammary gland ducts, and they were taken at 400 X magnification. Images on the top were taken at the time I did the initial staining, and the images on the bottom is those were taken after a certain period of time after initial staining. So June 2021, you can see pink and purple stain just fine in xylene. But when we look at pine oil, there's barely any pink, though you can see a little bit of red blood cells right here, slightly, only faintly stained pink. But for the most part, pink is absent from this mammary gland duct. The purple is still there though, which is good. 6 months later, the pink disappears from the xylene and pino group. This tells me that after 6 months that the eosin quality fades away after 6 months, at least for this group right here. Then we're moving on to December 2021 data. Here's the initial images, and then 2 months after December 2021. The colors still appear the same as the ones initially. So what can I conclude from figure 4? Well, it's actually too early to conclude. We don't know for sure if the age of the reagents are to blame for the fading of the pink in H&E staining, like what we see this data right here. It could also be because of the protocol that I made for this staining. And then the third part is what if it's because of pine oil being used in H&E? We don't know for sure yet. We need more data for the quality comparison of xylene and pine oil H&E staining over time. All right, and to summarize my project, we determined that pine oil is a suitable alternative to xylene for deperiodization steps and adequate for dehydration and clearing steps of histological staining techniques. Pine oil treated histological stains had color balance and color intensity issues as shown in my figures. And you can already see that eosin quality fades away at 6 months for both xylene and pine oil treated stains. But then once again, it's too early to conclude if it has something to do with the age of the reagents, my protocols, or if it's because of pine oil being used in the stain. The future directions for this project, one of them being to optimize both H&E and mesotrichum staining protocols with pine oil and testing other tissue types such as kidney and lung, since I only use mammary gland tissue for this project. Might as well test it on other tissue types. We would also like to test pine oil in other downstream histological applications such as immunohistochemistry. And then last but not least, recruit histologists to evaluate the quality of pine oil in histological stains. Here are my references, and I would like to give my acknowledgement to DODD and MSCIP for partially funding this project, as well as acknowledging other contributors to this project histology lab from spring 2021. As well as Bertha Garza, Onaja Bloom, and Gabrielle Vilbos. And thank you for listening. I will now take some questions.
Okay, your research is so freaking cool. Okay, so I have a question. So say that the like, you know, your samples like do start to fade like at 6 months, would that be a problem?
Yes, that is a problem because in histology, we would like to preserve these stains as long as possible. And by that, I mean, like, I want to preserve this for years to come. Like, what if I need to like reference this, like, let's say like, you know, like in like settings where like you want to like identify, like, is there something wrong with this patient or something? We want to like be able to like reference back, like, hey, this is what we're seeing in this patient and stuff like that.
Okay, and so if it were to fade, what would you use like instead? Like, or is that like where you're trying to figure out now? Because you want to replace xylene with pine oil, right?
Yes.
So if pine oil doesn't work either, like would you just go back to the xylene or would you replace that with something else?
I would actually like prefer not to use xylene because of how toxic it is, but pretty much I would like trace back my steps and figure out what did I do wrong that caused the fading to occur. So as I mentioned before, I don't know if it's because of how old the reagents were, or does this have to do with my protocol? I gotta like try all of that.
Okay.
And I also use pine oil because this is something I've been working on for a long time. A year.
Yeah.
Unfortunately, I'm graduating next month though, so I had to like wrap this up.
Congratulations to you. That's so good. Oh, that's so exciting. Yeah, well, you did really good research. You did a really good job. Your presenting skills are amazing. But yeah, thank you for this. And I really learned a lot. And I hope that your the pine oil doesn't fade. So when will you know if it fades or not?
Actually good news because since this poster was made back in February, I didn't get a chance to update this. Obviously, I had to reuse this poster for the SRC, but I look at the stains that I did back in December, which is this batch right here, right?
Uh-huh.
I look at that data and the pink is still there, so
Okay,
I'm
that's
good.
good. That's good.
And
Okay. Okay.
we're in April, so how many months has it been? Only 4 months since December, so yeah, the pink is still there. That's why.
Okay. Okay. That's good then. Congratulations.
Thanks.
Yeah. Yeah, so I'll let you go. Thank you once again for
Mhm.
presenting. You did an amazing job and good luck to you and congratulations on your graduation.
Thanks for visiting my booth.
Yes, of course. Bye. See you later.
Yeah.